Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, so in the previous videos what we've done is we've seen many examples of ion channels which we could say are proto-oncogenes, i.e. a gain of function in those proteins seems to be associated with an oncogenic phenotype. Okay, what we're now going to look at is two examples of ion channels which seem to be the opposite. They seem to be tumor suppressor genes, and it seems to be a loss of function in those that is necessary in order for oncogenic progression. Okay, right, so the first one we're going to look at is the KCNQ1 channel. Okay, and basically this is a homotetramer of 4 KCNQ1 subunits. Okay, and the other name for a KCNQ1 subunit is also to call it a KV7.1 subunit. So that's the more standard nomenclature for KCNQ1. However, KCNQ1 is the pervasive nomenclature. Okay, so this is the one that it would be nice if everyone used. This is the one that generally people do use. Okay, so just so that I've got a picture to point at, basically it's this common homotetramer that we've seen many times now, okay, sitting in the plasma membrane like so, made up of 4 identical subunits which are all KV7.1s, which you can also call KCNQ1. And the entire channel is then called a KCNQ1 channel, or a KCNQ1 homotetramer, or KV7.1 homotetramer, whichever you prefer. Okay, right. So basically, you find that in the early stages of neoplasia in the colon, okay, that KCNQ1 has a high expression, and in the later stages it has a lower expression. So let me just get my picture back again. So here is the picture in which I explain the progression of cancer in terms of the formation of these different clones and the gradual evolution of these clones. Okay, right. So what I'm saying is in the initial clones, the early clones, they have quite a high expression of KCNQ1. And then in the later clones, the later stages of neoplasia, where we're going towards malignancy, when we're going towards cancer, these suddenly have a lower level of KCNQ1. Okay, now what does that suggest? Well, remember, as you go along the clones, each of the clones is getting better at surviving. It's getting better at producing a massive, great population of cells. Okay, so if this is a finding that we're finding commonly, then it suggests that potentially the lower expression of KCNQ1 might be providing the cells with an advantage. Okay, and therefore potentially one of these mutations that you're getting that is advancing the clone forward might be some loss of function mutation in KCNQ1, in this case something that results in its expression going down. Okay, potentially a mutation in its promoter that stops it being transcribed. Okay, that would then lead to reduced KCNQ1 expression, and then if KCNQ1 was halting proliferation, at least to some degree, then removing that brake would suddenly allow proliferation to go at a rate that even before it had not reached. Okay, and that would provide the cell with an advantage. Okay, so that is potentially a role of KCNQ1 as a tumor suppressor protein in these colorectal cancer cells, or at least these colorectal neoplastic cells. They're not quite at cancer stage yet, okay, but they're progressing towards the cancer stage. Okay, right, so that's KCNQ1. Let's now talk about another example of a tumor suppressor ion channel potentially. Okay, so this is an example from the TRP channels. Okay, so the example that I have here is the channel TRPM1. Okay, so we've already looked at TRPM8, the cold sensor. TRPM1 is another homotetramer of 4 TRPM1 subunits that again forms a non-selective cation channel. Okay, now it's believed to act as a tumor suppressor in melanoma progression. Okay, so in melanocytes. Right, so the idea here again is basically that in the early stages of neoplasia in melanocytes, you have a high expression of TRPM1, And then in the later stages of neoplasia of melanocytes, you have a low expression of TRPM1. And this suggests potentially that some loss of function mutation in TRPM1 that affects its expression has occurred, and that now, without the break of the TRPM1 channels that the TRPM1 channels provides, the cells are now proliferating at a level that they weren't before, and that promotes the oncogenesis, promotes the progression of the clone to a more deadly clone towards the cancer clone. Okay, so TRPM1 and KCNQ1 both seem, therefore, to potentially be tumour suppressors. Okay, how they have an anti-proliferative effect, we don't know. Okay, this is, no pathways are here, it's just raw findings at the moment. Okay, right, so that's now the tumour suppressor ones discussed. What I now want to move on to is how ion channels are potentially involved in the metastatic phenotype. Okay, so how they're involved in cell motility. Okay, and for this I need to tell you a little bit more about how cells actually move. Okay, so when we were originally discussing the development of cancer, we did talk about epithelial mesenchymal transition and then mesenchymal epithelial transition, but we didn't actually talk about how the mesenchymal cells themselves actually move. So I now want to discuss how the cells actually move. So let's draw a cell here. So let's say this is our cell and we want this cell to move. So how does it occur? Well, basically what happens is the cell is going to produce a process known as an invadopodia. Okay, so let me show this occurring. So what's going to happen is, let's say the cell wants to move to the right. Okay, so it wants to move to the right. And this arrow isn't meant to be showing which direction it's going to move in. This is meant to be showing one picture going to the next. Okay, so now what's going to happen is the cell that wants to move to the right is going to make a little process on its right-hand side, like so. Okay, and this little process is going to squeeze into the new space that it wants to move to, and this process is what's known as an invadopodia. Okay, now, very importantly, you have to understand that the space in between cells is not just an empty space, okay? It is in fact a meshwork of a huge number of molecules. Okay, so let me just draw this out. So, if we have cells here, this space between cells is not just an empty space. Instead, you have molecules all over the place, making up a meshwork everywhere, a meshwork of connective tissue, loads and loads of collagen, but also other molecules besides collagen, okay, in red there. Okay, and this meshwork of molecules, this is known as the extracellular matrix, the ECM. Okay, so this stands for extra, that's the E, cellular, that's the C, and then M, that's the matrix. Okay, right. Basically, if you want to move, you have to move the extracellular matrix out the way. So when this invadopodia extends into a space, it's going to be breaking down the extracellular matrix around it to project into that space. Okay, right. The next phase then is that the invadopodia is going to expand. Okay, so I might draw this in dashed lines. So the invadopodia is going to start expanding like that. And the back of the cell here, and the special terminology that's quite helpful here, this end of the cell, which is the invading end of the cell, this is known as the leading end of the cell. Okay, and the portion of the cell that isn't, is moving away, okay, this portion of the cell here, that's in the opposite, at the opposite pole of the cell effectively, this is what's known as the trailing end. Okay, right, the leading end, basically, where the invadopodia is at, is going to expand, whilst the trailing end is going to decrease in size, basically. It's going to contract back down. And as you can see, if we continue that process up, we're effectively going to move the entire cell forward. Okay, so if we expand this side even more, up to this, and we take this right back down here, effectively the entire cell has now moved to the right, basically. Okay, and that's what's going to happen. So let me draw a little bit of this. So I'll draw that first step there. So now the invader podia has swelled out like so, and the trailing end has reduced in volume slightly. Okay, so I've said the key word there, volume. It's all about changes in cell volume. Now, we have seen how ion channels can mediate changes in cell volume already when we looked at eferagogo-2 and its role in pre-mitotic condensation. So this might be ringing bells that potentially ion channels are going to be involved here to produce these changes in volume at either end. It's also worth noting that in order for that invader podia to expand out at this end, of course it's going to have to continue breaking down extracellular matrix to produce that space for it to expand. And of course what this will eventually go to is just the whole cell now moved to the right, basically. Okay, like so. So that's how cells move. They first make this little invader podia, which then swells whilst their trailing end decreases. Okay, right, so let's now talk about the role of ion channels in this entire process. So the first thing to say is that calcium signaling actually becomes important first, and then calcium signaling is going to affect the ion channels, especially at the trailing end. So basically, in these cells that are moving, you have a calcium gradient across the cell. So let's have a look at this cell here. Basically, calcium concentration is higher at the trailing end, and then it gets lower towards the leading end. So you have a gradient where you have high calcium here, so I'll draw big calcium at this end, and then very little calcium at this end. Okay, so you establish this intracellular calcium gradient. Okay, God only knows how, but it's there. Okay, and this plays, or is believed to play, an important role in actually making this trailing end decrease its cell volume. Okay, specifically because the high calcium is going to activate calcium-activated potassium channels. Okay, which I'll draw here. So this ion channel on the trailing end here, this is going to be an intermediate conductance calcium-activated potassium channel. So this is an IK channel. Okay, and the high calcium in the cytoplasm at the trailing end of the cell is going to then lead to this intermediate conductance calcium-activated potassium channel opening. Potassium ions will then leave the cytoplasm of the cell and go into the extracellular fluid. Okay, and just like before, this is now going to distort fluid volume homeostasis over here. Okay, now again what's going to happen is chloride channels, which are always open and which allow the chloride to passively distribute. Chloride is going to follow the potassium ions that have moved out. Okay, as I say, because chloride is passively distributed. Okay, so to maintain electroneutrality, the chloride is going to follow the potassium. And again, this chloride channel is believed to be ClC3. Okay, then you have overall now moved salt, you've moved potassium chloride. Okay, so you've now established an osmotic gradient across that cell membrane, and water is now going to move out of the cell. Okay, so let me put this here. So I will colour this in red here. So again, the water is going to move via aquaporins. That little red blob there, that's supposed to represent an aquaporin. Okay, so the water is now going to follow, and that's going to decrease the volume in this trailing end of the cell, and therefore that's how the trailing end of the cell sort of wastes away, atrophies away is probably a nice word to put it. Okay, right, now just for a complete picture here, I'm going to tell you how at the invader podia end, what happens to increase the cell volume over here. Okay, so at the invader podia end, what's believed to underlie the increase in cell volume is the presence of an NKCC, a sodium-potassium chloride cotransporter. Okay, so this stands for sodium, that's the N, potassium, that's the K, and then chloride, that's the first C, and then it's cotransporter, that's the second C. Okay, and basically this transports one sodium ion in, okay, one potassium ion in, okay, and then two chloride anions in. Okay, right. And it uses the electrochemical gradient of sodium to drive that movement inwards, basically. And obviously this is moving a lot of salt into the cytoplasm of the cell. Okay, so what now can happen is water can follow through an aquaporin again. So I'll just put one of these aquaporins here, which can bring the water in, basically. Okay, and that's how the volume within the invader podia can increase and then cause the engorgement of the invader podia whilst the trailing end atrophies away. Okay, right, so clearly then, the IK potassium channels are very important here. Also those ClC3 chloride channels are very important in this process. And in fact, you can block the movement of cells. Specifically, I'll tell you about a specific type of cell that this has been done in. It's been done in what's known as an MDCKF cell. Okay, not in like that. MDCKF cell. Okay, so it stands for something silly like Made in Derby canine kidney cells. Okay, and then this F just denotes the fact that they have been transformed. So basically what this is it's a renal tubular cell. So a renal epithelial cell, one of those cells that lines the renal tubules. Okay, and it's been transformed, which has been transformed. Okay, so it's cancerous. Okay, now these cells show a lot of motility. Okay, however, you can actually inhibit their motility by stopping this atrophy of their volume at their trailing end. Now, from the model that I've given you, how could you stop this atrophy of their volume at their trailing end? Well, by blocking that intermediate conductance calcium-activated potassium channel. Okay, and it turns out there is a very effective toxin that I believe is a scorpion toxin, which is capable of blocking that intermediate conductance calcium-activated potassium channel. And this is what's known as charybdotoxin. Okay, so charybdo, and I'll just move this up a little bit, charybdotoxin. Okay, so charybdotoxin is capable of blocking the intermediate conductance calcium-activated potassium channel and is actually capable of blocking migration in these MDCKF cells, which are transformed renal epithelial cells. Okay, right. Potentially another thing to note, but which isn't understood at all, is that there is actually another ion channel expressed exclusively in the invader podia, and it's not really known what it does at all. Okay, and it's an inward rectifying potassium channel. Specifically, it's a KIR 4.2 inward rectifying potassium channel homotetramer. Okay, so I'll just put that in here. Oh, actually, I won't. It's not going to fit in there. Okay, so you find these KIR 4.2 homotetramers present then in the invader podia, and as I say, it's not understood at all what their role is. It's thought that they probably have non-canonical signaling roles. Okay, so signaling roles that have nothing to do with their ability to conduct potassium ions. Okay, right, and they're believed to probably have something to do then with the motility process. Okay, right, so whilst we're on the subject of motility, I'm going to mention a few more ion channels that are important in motility in cancer cells and seem really important in metastasis in certain forms of cancer. Okay, so the next one up that I want to talk about is what's known as the HERG channel, which is an extremely famous channel in pharmacology. So what does this stand for? This stands for the human, and then it's ether-a-go-go, which we're used to by now, ether-a-go-go, and then it's related gene. So this is the human ether-a-go-go related gene. And basically this is a channel that is a homotetramer of 4 HERG subunits. Now the more sensible name for HERG subunit is a KV11.1 subunit. So remember, the ether-a-go-go potassium channel subunits were KV10.1 and KV10.2. Now this human ether-a-go-go related gene is KV11.1, so they're near each other in the naming system here. Okay, right. So again, it's a homotetrameric voltage-gated potassium channel, as we've seen many times before. Now, the reason HERG is so famous in pharmacology is because of how important it is in the heart. It is absolutely incredibly important in the repolarization phase of the cardiac action potential. Okay, and it's one of the most famous anti-targets in drug discovery. Okay, so basically one of the key reasons that a lot of drugs fail ever to get to the market is because they have they can have extremely dangerous side effects, okay? Because many drugs that are meant to do completely different things end up actually blocking this annoying potassium channel in the heart, okay? And what it causes is something known as long QT syndrome, where the cardiac action potential becomes much longer because now it takes much longer to repolarize the electrical potential difference across the cardiomyocytes when this channel is blocked. Okay, and this can then lead to dysrhythmias, okay, and dysrhythmias can then lead to cardiac arrest, basically, the heart stopping beating, and that's obviously very fatal if you're not in hospital extremely quickly. Okay, so cardiac arrest. In fact, if you're not already at an operating table, it's very difficult to do anything about that. Okay, right. So that's one of the reasons many drugs actually fail, because they lead to dysrhythmias in the heart because they block the human effragogo-related gene potassium channel, okay, and cause, therefore, fatal cardiac arrests. Okay, so it's a renowned anti-target in pharmacology. In fact, I think every drug is now checked very early on in the drug discovery process to make sure that it does not block the human effragogo-related gene potassium channel. Okay, so that's where it's usually important. However, it's believed also to actually play a part in the metastatic phenotype in some leukemias. Okay, so it's believed to play a part in leukemias. So firstly, let me just explain what a leukemia is. So a leukemia is actually a cancer that occurs within the bone marrow, okay, and it occurs within the cells which make certain white blood cells, okay. And when you get that tumour of these precursor cells for making white blood cells, what then ends up happening is that these cells end up still making white blood cells. So effectively, if I draw a picture here, let's have the bone here, okay, like so. And then what's going to happen is you're getting this tumour of these precursor cells within the bone marrow, okay, and basically some of these cells will end up differentiating into white blood cells and going into the blood. So you end up with these cells within the bloodstream. Okay, I just closed the window. Okay, so that's what leukemia overall is. It's this cancer within the bone marrow of precursor cells to white blood cells, and you end up with certain of these cancerous cells ending up in the blood, basically. Okay, now, human effragogo-related gene is believed to be extremely important in the metastatic properties of these leukemia cells, so their ability to actually invade peripheral sites and set up secondary tumours. Okay, indeed, what human effragogo-related gene is found to do in these leukemia cells is that it forms a complex with certain other proteins, and specifically the other proteins that it seems to form a macromolecular complex with are the beta-1 integrin and also the vascular endothelial growth factor receptor 1. So let's just draw a picture for this. Here is our human effragogo-related gene potassium channel. Okay, then we'll have the beta-1 integrin here, maybe. Okay, and then also the vascular endothelial growth factor receptor, which again is a single membrane spanning protein. So both the beta-1 integrin here and also the vascular endothelial growth factor receptor 1 are both single membrane spanning proteins. Okay, there we go. And this complex, for some reason, seems to be extremely important in the metastasis of these leukemia cells, okay? So without this complex, you have problems if you're one of these cancer cells metastasizing. They don't seem to be able to invade at peripheral sites once they're in the blood and then actually set up secondary tumors. Okay, so there's interest in whether we can target this complex, break it apart, and stop the leukemia cells hence from metastasizing to peripheral sites. Okay, right. On a similar theme, there is also a complex that has been found between a calcium release activated calcium channel, a crack channel, and also a small conductance calcium activated potassium channel. Okay, so let me just discuss this over the way. Okay, so basically, let me explain firstly what a crack channel is. Okay, so a crack channel is a calcium release activated calcium channel, and this is involved in a phenomenon known as store operated calcium entry. So basically, store operated calcium entry is all about the fact that when you release calcium from intracellular stores, such as the endoplasmic reticulum, okay, you are going to get calcium depleted from the intracellular stores. Okay, this calcium has to be replaced from somewhere, and it's gotten from the extracellular fluid by a process known as store-operated calcium entry. Now, the channels that open in order to allow that calcium from the extracellular fluid to come into the cytoplasm before it can then be loaded into the endoplasmic reticulum is the calcium release activated calcium channel, which is known as the crack channel. Okay, right. Now, this is made up of a homotetramer of 4 proteins that are called aura-I proteins. So let me show this here. So here is our calcium release activated calcium channel. Okay, and it's made up of 4 separate subunits here, which are known as aura-I1 proteins. So this is an aura-I1 protein. And let me just show you the structure of an aura-I1 protein. So this is the membrane spanning topology of an aura-I1 protein. You have the amino terminus intracellularly, along with the C-terminus, and then you have those 4 membrane spanning alpha helices. Okay, so you put 4 of these aura-I proteins together to make a calcium release activated calcium channel, and it's important for bringing calcium in from the extracellular fluid into the cytoplasm so that calcium can then be loaded back into the endoplasmic reticulum. Now, where has this been found in cancer? Well, it's been found to be complexed with another ion channel in certain breast cancer cells. And it seems again that this complex is extremely important for the metastasis properties of these breast cancer cells. Okay, so let me show this complex. So the complex seems to be a complex between the aura-I crack channel, okay, so this aura-I1 homotetramer here, okay, and then also a SK potassium channel, okay, specifically an SK3 calcium activated potassium channel, okay, which remember is a calcium activated potassium channel made up of 3 KCA 2.3 subunits, so KCA 2.3 subunits. Okay, so this complex seems to particularly form in special portions of the membrane known as lipid rafts, which are portions of the membrane which have very high cholesterol levels, okay, and also high sphingolipid levels, okay. So, there is interest in whether disturbing this complex, potentially by disturbing lipid rafts, could stop metastasis of breast cancer cells. Okay, right. So that now concludes our discussion of cancer and ion channels.